Okay, so the last time we had talked a bit about gluconeogenesis, and I wanted to talk a little bit more about that. So as you recall, this is actually kind of a nice diagram. On page 552 of your book, you see here gluconeogenesis and glycolysis, plus you also see the way in which they're regulated. So red means that it's going to be a negative regulator, green means there's going to be a positive regulator. As I said, for glycolysis, there are 3 points of regulation: hexokinase, which we just talked about, phosphofructokinase, and also pyruvate kinase. So those are the 3 major places. And I said energy charge will regulate these activities. And then on the right-hand side, you've got gluconeogenesis, and you've got controllers there as well, places of control. And the controllers are going to be the bypasses. So this is bypass number 1 here, these 2 enzymes in red in the very bottom. Bypass number 2, where you have fructose 1,6-bisphosphatase, and bypass number 3. All right. So let's talk about these 3 bypasses. So I'll go through the reaction mechanisms. Bypass number 1. Where you have pyruvate going to phosphoenolpyruvate. Bypass number 2, where you've got fructose 1,6-bisphosphate, and that's going to be going to fructose 6-phosphate, and then bypass number 3, where you've got glucose 6-phosphate going to glucose. So those are the 3 bypasses, and we call them bypasses again because they use different enzymes than what's used for glycolysis. Okay, the first bypass is the most complicated. The second and third are very, very simple. So let's talk about the first bypass. It's 2 steps. In the first step, we've got pyruvate plus HCO3 minus plus ATP, and that's going to go to oxaloacetate plus ADP plus inorganic phosphate. Anybody remember what we call that? What I just underlined. Think way back to your general chemistry. Close. It's not carbonic acid. It's very close though. It's the deprotonated form of carbonic acid. Anybody know? Starts with a B. Bicarbonate. Okay, bicarbonate. Okay, bicarbonate. So this is bicarbonate, ATP. Oxaloacetate has 4 carbons on it, just to let you know. And you should probably really understand its structure, maybe even memorize its structure. It's not that hard. It's 4 carbons long. Okay, how does this work? In the first step, we need to activate this bicarbonate so that it has a good leaving group, and therefore pyruvate will be able to attack it with ease. Okay, so let's talk about the first step. I'll write out bicarbonate. So that's the structure of bicarbonate, okay? And then we've got ATP. I'm just going to write this in an abbreviated form. So that's the terminal phosphate, the gamma phosphate, okay? And that's going to be attached to ADP. So altogether, this is ATP, by the way, okay? And we just do simply a nucleophilic attack right there. You guys have probably seen this dozens of times now. and the ADP will leave, and now we have a phosphorylated carbon there, carbonate, okay? And we used to call this carbamoyl phosphate. I think your book may call it something else, but it's called carbamoyl phosphate. Let's go ahead and write that structure. ADP is going to leave. Okay, now this phosphate is a really good leaving group, okay, so that will be used eventually. Now I have to introduce to you another cofactor, and the name of this cofactor, we've talked about a lot of cofactors, but the name of this cofactor is called biotin. Biotin, okay, and biotin looks like this. And this is part of the enzyme that has a side chain known as lysine. Okay, so that's the lysine side chain. Okay, it's been modified. Okay, and so this is covalently attached to this enzyme. By the way, the name of this enzyme is called pyruvate carboxylase. So I'll write the name of the enzyme here. Pyruvate carboxylase, and it is a synthase, not synthetase, but it's a synthase. Okay. So how does this work? First of all, this proton leaves. Okay, so you have some kind of an acid group. A base that's going to take off that proton. Okay, and so we create. I'm just going to write, this is the business end of the molecule, the business end of the molecule. So now we've got some electrons there that are ready to attack. And this will attack. Sure what happened here. That activated carbonate, and so the phosphate is going to leave, and so now we get biotin attached to the carboxyl group there. So we'll go ahead and redraw that. Okay, so that is the biotin attached to the bicarbonate. And then the next step, we need to attach this to pyruvate. And so pyruvate looks like this. And so what happens is that a base, just go ahead and redraw this a different way. So that's pyruvate. So a base is going to come along, remove that proton there on the methyl group, okay? And then you get a double bond formed, a carbene bond, and then the oxygen, the double bond oxygen is going to become single bonded. Okay. And then finally, in a concerted reaction, this is going to make a double bond here, and this is going to attack right here. We recreate our biotin, and now we're going to get a carboxyl group placed on the end of this pyruvate. And this is oxaloacetate, which we sometimes abbreviate as OAA. Okay. So that is the first step of bypass number 1. The second step is much, much easier. In the second step, let me go ahead and write out the reaction for you. It's going to be OAA plus GTP. I'll write down here second step. And that's going to make phosphoenolpyruvate plus GDP plus CO2. Okay. How does that work? I'm going to just draw out GDP with a terminal phosphate here, so that's going to be actually GTP. I did the same thing with ATP. So that actually is GTP. Okay. I'm getting this ready for attachment to the oxaloacetate. I'm going to draw out oxaloacetate. Let's see how I should do this. Oh, by the way, I misdrew this oxaloacetate. Let me just go back up here. It's missing a carboxyl group. Okay. 1, 2, 3. 1, 2, 3, 4. Okay. Sorry about that. So that's oxaloacetate. Okay. So next, how am I going to do this? I'm going to redraw oxaloacetate. By the way, see this little blinking light down here? Right here? There's like this blinking light. That's like the edge of the video. So I'm trying to make sure that I don't like go to the right so that we can capture everything on the video. So I'm constantly looking at that green light to make sure I don't go too far to the right. Okay, next. We're going to have this reaction here. Okay. Okay. 1, 2, 3, 4. So that's the reaction that's taking place here. What's going to leave is CO2. This is the CO2 part right over here. Okay. And what we're going to create is phosphoenolpyruvate which will look like this. And that's phosphoenopyruvate. I didn't write out the whole phosphate. That PI is the entire phosphate there. Okay, so we are done with bypass number 1. I've showed you that we have to activate the bicarbonate, we have to use biotin, and I told you that was a synthase reaction. And then in the second step, we're actually removing CO2, and we're making phosphoenopyruvate from oxaloacetate. By the way, this reaction up here, pyruvate carboxylase, requires either magnesium or manganese. Anybody want to guess why? Why does it require a metal? Excuse me? To catalyze. Okay, very good, very good. But specifically, where do you think the metal's going to be involved? Okay, it could be the carboxylic acid. The prevailing concept is that these are divalent, and they actually help to stabilize the wow, I guess some of my writings have gone away here. That's too bad. Okay, they stabilize the nucleic acids here. So the ATP and the GDP and the GTP, those are what they're responsible for. So you guys can go ahead and see that in the book, but they're responsible for helping to remove negative charge from the terminal phosphate so that the terminal phosphate becomes a good electrophile. Okay. Overall, bypass number 1, the overall reaction is pyruvate plus ATP plus GTP. It's going to give us phosphoenopyruvate plus GDP plus 2 inorganic phosphates. So we'll get 2 inorganic phosphates from actually, we're just going to get 1 inorganic phosphate. That's wrong. 1 inorganic phosphate. Okay. Next, the PEP is going to exit the mitochondria. Okay. And the PEP then will get converted into glucose through gluconeogenesis. What are those steps? Those steps are pretty simple. Here's bypass number 2. We have fructose 1,6 bisphosphate plus H2O, and that's going to create fructose 6-phosphate. I'll just abbreviate that as F6P plus an inorganic phosphate. Can anyone guess what class of enzymes this would belong to? Fructose 1,6 bisphosphate going to fructose 6-phosphate and inorganic phosphate. And I'm using water to do this. Hydrolase, very good. So this is a hydrolase. Very good. Okay, because hydrolase will be severing that bond. Okay. Bypass number 3. we have glucose, 6-phosphate, plus water is going to go to glucose plus inorganic phosphate. We want to guess what class of enzymes would carry this out. Hydrolase, very good. So this is another hydrolase. And then finally we end up with glucose. Okay. The liver, as I said before, is responsible for a lot of gluconeogenesis because during times of starvation in the body, liver is responsible for generating glucose in order to deliver that to other organs in the body. And the liver, just draw a liver here. There's my liver, okay, right? And the liver does this in two ways, delivers glucose. It can go through gluconeogenesis, which I just talked about. It can also do something called glycogenolysis. Okay, so we've talked about gluconeogenesis. Okay, there's another way that it can make glucose, and that's through a process called glycogenolysis. Okay, glycogen is a polymerized form of glucose. Okay, lysis means that it's going to get lysed. Okay, and this breaks down large, complex molecules like glycogen in order to produce glucose. Okay, so that happens in the liver, and that glucose then will be exported to other organs in the body, the brain, the heart. Oh, that's nice. Okay, and then we've also got the muscle. Okay, so we got all those organs that need liver in order to do that. And of course, there are hormones that do this, and one of the hormones is glucagon. So we talked briefly about glucagon and how that signals starvation, or when you're sleeping at night for like 8 hours and you're close to morning, the glucagon comes in and causes the liver to do this. So one of these is gluconeogenesis, the other one's glycogenolysis, and the idea is to make sure that these other organs survive, especially the brain. The brain is very, very dependent on glucose. I'm going to talk a bit about how the energy charge controls glycolysis and gluconeogenesis. I'm going to make a table here. Okay. And the table is going to have 4 columns. You got the molecule. I'm going to say the effect on EC, energy charge. I'm going to say the effect on glycolysis. I'm going to say the effect on gluconeogenesis. Okay. So here we go. AMP is the first molecule I'll introduce. Its effect on energy charge, if you think of the energy charge equation, AMP is found in the denominator. So if you have more AMP, your effect on the energy charge is going to go down. Okay? And your effect on glycolysis is that it's going to increase the activity of PFK1. That stands for phosphofructokinase 1. That's the enzyme that's found in the second regulatory step of glycolysis. Okay, so it's going to increase that activity. Therefore, you're going to have more flux, metabolic flux, through glycolysis. Also, it's going to decrease fructose 1,6-bisphosphatase. Okay, so it's going to decrease that. This enzyme is found in gluconeogenesis, and it's bypass 3. Okay, what about ATP? ATP, as you can imagine, if you have increased levels of ATP, that's going to increase your energy charge. Okay, so the numerator is going to increase in that equation for energy charge. Okay, the effect on glycolysis is to decrease phosphofructokinase 1, PF, PF. Okay, I have trouble spelling and talking at the same time. PFK1. And it's also going to decrease pyruvate kinase, which is the 10th step in glycolysis. So both of those will decrease. Okay? And there's no direct effect on gluconeogenesis. Okay? No direct effect. Gluconeogenesis. I'll just say no direct effect. Okay, what about another molecule? What about ADP? Whoops, no, okay. ADP. ADP, its effect on the energy charge is to decrease it, okay? So you have more ADP, you're going to have a decrease in energy charge, okay? You're going to increase PFK1 activity, and it has no direct effect on gluconeogenesis. Here's a couple more that you should know about. One is called citrate, which we will talk about when we talk about the citric acid cycle. I really don't know what its effect is on the energy charge, but I do know that it regulates phosphofructokinase. So it's going to increase phosphofructokinase 1 activity. No direct effect on gluconeogenesis. Another molecule which we did talk about, acetyl-CoA. Acetyl-CoA will decrease the activity of pyruvate kinase, okay? And it increases the activity of pyruvate carboxylase. Pyruvate carboxylase. Does anybody remember the name of the cofactor that's used in pyruvate carboxylase? Anybody remember pyruvate carboxylase? Biotin. Just went over it, right? So pyruvate carboxylase, it puts a carboxyl group onto the pyruvate and makes it oxaloacetate. Biotin is the cofactor used for pyruvate carboxylase. Okay, and so that's that. And then finally, the last one that I need to talk about is this thing called fructose 2,6-bisphosphate. And its effect on glycolysis is to increase phosphofructokinase 1. I'm sorry here. Let me just change that. That should be an F. Okay. And it's going to decrease fructose 1,6-bisphosphatase. Fructose 1,6-bisphosphatase. phosphatase. Okay. My purpose of showing you this is to give you a general idea of a few things. Yes?
For the second to last one, is it PK or is it supposed to be PF?
The second to last one is PK. That's pyruvate kinase.
That's citrate lowered PFK.
Citrate. I misread my notes. I appreciate that correction. Thank you very much. You're exactly right. It lowers PFK1. Okay, let me check these again with my notes. Boom, boom, boom. Yes, citrate lowers PFK1, and acetyl-CoA lowers pyruvate kinase activity, and fructose 2,6-bisphosphate increases phosphofructokinase 1. Thank you very much. I appreciate that. What I want to show you in general here, energy charge is important. Okay, and energy charge, as I said before, you have high levels of energy charge. That means you want to basically do anabolism. Okay, so you're going to increase gluconeogenesis in many of your cells. So you're going to be, I'm going to talk about some exceptions here, but in most of your cells, you're going to increase gluconeogenesis as well as other anabolic pathways. If you lower the energy charge, if you're down on energy, if you don't have enough energy, and your ATP levels are low relative to AMP and ADP, then you're going to shut down your anabolic pathways, and you're going to increase your catabolic pathways. One of these is glycolysis. And there are these other effectors as well that will bind to allosteric sites on these proteins that I suggested. So there are these major enzymes that are regulated by glycolysis and gluconeogenesis. And that means that we will not have a futile cycle. We will not have both glycolysis and gluconeogenesis running at the same time in the organ. I'm just going to draw for you the structure of fructose 2,6-bisphosphate. Then I'll let you guys go. So this is fructose 2,6-bisphosphate. And there's not very much of this in the cell. This is not an intermediate in glycolysis or gluconeogenesis because it's fructose 2,6-bisphosphate, not fructose 1,6-bisphosphate. This was discovered relatively recently, and it is an allosteric regulator of glycolysis and gluconeogenesis. I want to mention the numbering system here. So this is number 1, this is number 2, this is number 3, 4, 5, 6. And so you could see that on the 2-carbon, you have the phosphate attached to this. This is why this is called fructose 2,6-bisphosphate. We will finish up chapter 13 next time, and we will probably get through most of the citric acid cycle on Friday. Okay, you guys have a wonderful day. I'll see you on Friday.